First of all, what we have? The nucleus. So nucleus, they have nuclear pores, and the nucleic membrane is double membrane like this. So here the nucleus is present. Inside the nucleus, what happened? Transcription, mRNA is formed and ribosomes are formed by the nucleolus. Then what happened? This nuclear membrane, the outer one, is always connected extensively to the RER. So the RER is there. Very extensively they are connecting like this. RER is there, right? And on the RER, what we can find? Ribosomes. Ribosomes are present. So the nucleus is there, RER is there, ribosomes are there. Now what happened? The mRNA which is coming to these ribosomes and the protein translation occurs. After the translation occurs, they will be processed inside this RER. Now they are processing. Primary folding, that means first ever foldings. Primary processing starts to happen. After the primary processing is done, what happened? Parts of or ends of this RER will be broken off with the processed proteins and they are moving towards the Golgi. So we have Golgi. What happened? These parts, they come and join to the Golgi, and actually they are the one who makes the Golgi. They are the one who makes the Golgi, right? Like this. They come and join and makes the Golgi. Inside the Golgi, further modification occurs. Further modification means addition of carbohydrates, further folding, 3D shape of the protein, everything happens inside the Golgi. So when the protein is trying to leave the Golgi, that's completely functional protein. How they are going out is depends. So if the protein which are modified are hydrolytic enzymes, they will be turning into lysosomes. They package in the lysosomes. If they are just proteins, they will be packaged into the vesicles. They will be packaged into the vesicles. Now these guys are having two options. Either these proteins needed to be used in the cytoplasm. So here is cytoplasm, right? Here is the cytoplasm. So these two proteins, one option, first option is they can be used in the cytoplasm. So what will happen? Especially these vesicles, they just come, they split it and release the protein out. Can be used, no problem, right? But the lysosomes containing hydrolytic enzyme, that's a problem. Why? If they just broken off, what happened? Hydrolytic enzyme comes to the cytoplasm digesting everything, right? Therefore, most of the time what happened, these lysosomes, they just keep the hydrolytic enzyme inside. But just imagine we have a dead or half dead, let's say mitochondria is there. Mitochondria is there, not working properly. Now what happened? This lysosome goes towards the mitochondria which is half dead and break the to release the enzymes onto the mitochondria. Now what happened? Mitochondria will be digested. So it can be absorbed back to the cell. Nutrients can be absorbed back to the cell. So that's what I said. Lysosomes are involving in recycling the nutrients. So breaking down these hydrolytic enzymes breaks down the dead cell organelles. Sometimes, for example, let's say this is a kind of cell that secrete the digestive enzymes to outside. Like the cells in the stomach, they are secreting enzymes to the outside, right? Then what happens? These lysosomes will be moved into the cell surface membrane and fuse with it, releasing the enzymes to outside. So they can digest outside of the cell. They can digest outside of the cell, right? So until that they keep it inside the lysosome. But very interesting thing is this. Cell death. Cell death. How a cell can be dead? Because if all the nutrients are there, all the organelles, they can be made by themselves. Like mitochondria, chloroplasts, they can replicate by themselves. If one is dying, they can make a new one. And all other organelles also can be made by the cell. There is no reason cell to be dead, right? There's no reason. So that's a kind of mystery actually. If we can find out what is the reason cells are dying, we can live forever. Basically, immortality we call, right? We'll talk about that later. But when the time comes, just imagine there's an alarm. Okay, this day, for example, all the red blood cells, they are dying within a month. Like that, each cell, they have a lifespan. When the lifespan come, okay, alarm is running out. Then what happened? All these lysosomes comes into the cytoplasm and they burst themselves. That means all these lysosomes with the hydrolytic enzymes, they come to the cytoplasm and they just burst themselves. What happened? All the enzymes are released to the cytoplasm, self-digesting the entire cell. Suicide. They are suiciding. That's how cell death occurs. That is called apoptosis. Okay, so that's how all our cells are dying, and that's how we are getting older. Lysosomes, due to some reason, don't know, due to some reason, bursting by themselves and digesting the cell itself. That's how each cell is dying. So we are getting older. So if somehow we can manage to not to do that, we can live, right? So the thing is, you need to find out who is telling the lysosome to burst. What is the reason? What is that alarm that tells the lysosome, okay, now is the time, blast yourself, like that. If you can skip that or postpone that, then you can live.